Hello, biology enthusiasts. Welcome to this insightful practical session where we explore three key biological sciences practical. If you are preparing for exams or just love biology, this video will be super helpful. Let's dive right in. I'll start as usual with the confidentials. So in question 1, we required 20 mils of liquid L. We are going to come and see how liquid L was being prepared. A white tile, 3 droppers, 5 test tubes, a test tube block, a filter paper, tap water, or water in a wash bottle. Means of heating: Bunsen burner, portable burner or spirit burner, water bath, etc. Means of stirring: glass rod, iodine solution, Benedict solution, 2% sodium hydroxide solution, and finally 1% copper sulfate solution and a 30 centimeter ruler. Guidelines for the preparation of liquid L. You are going to add 50 mils of distilled water and there you add 10 grams of starch. Or in other words, I'm saying to 50 milligrams of distilled water, add 10 grams of starch. Heat while stirring to dissolve the starch. Allow to cool. To the solution, dissolve 20 grams of glucose. To the solution, add 20 mils of egg albumen or freshly obtained raw egg white. Stir the mixture thoroughly, and to it you add enough distilled water to make 100 mils. For question 2, this one was in the question paper, and it was an electron micrograph showing a cell and parts of neighboring cells in a tissue of a plant. Question 3, you had photographs of 10 specimens of invertebrates, where a photograph of a mollusk was labeled E, a photograph of a crayfish was labeled F, a photograph of a scorpion labeled G, a photograph of an earthworm labeled H, and a photograph of tapeworm labeled J. This question 3 can also be done by you having the actual specimens. Let's go to the questions. In question 1, where you are provided with liquid labeled L, using the materials provided, identify the food substances present in the liquid, record the food substances being tested for procedure, observations, and conclusion in the table below. So that is the table. Food substance, procedure, observations, and conclusion. B. After stirring thoroughly, place 20 drops of liquid L in test tube and boil it. Record your observations. What conclusion can be drawn from your observations in B1 above? Question 2. Name the parts labeled P, Q, R, S, and T. Name the kingdom to which the organism from which the tissue was obtained belongs. Kingdom. Give 4 reasons for your answer in B1 above. Measure the diameter of the cell in centimeters between X and point Y. If the magnification of the cells in this micrograph is 30,000, calculate the actual diameter of the cells in micrometers. Question 3. During a visit to a museum, students were shown 10 specimens of invertebrates on display. The teacher provided a dichotomous key shown on the next page to enable them to place each specimen on display into its taxonomic group. 5 of the 10 specimens that were on display are shown in the photograph below. So those are the specimens. All right. Those are the specimens. All right. We have E, F, G, H, and J. Use the dichotomous key to identify the taxonomic group for each of the 5 specimens shown in the photographs. In each case, show in the sequence the steps 1A, 2A, 2B, 5A, or 7B in the key that you follow to arrive at the identity of each of the specimens. So this is the dichotomous key. Animal with flattened body, go to 9. Animal without flattened body, go to 2. Animal with the body in shell, mollusca. Animal without a shell, go to 3. Animal with a segmented body, go to 4. Animal with the body not segmented, nematoda. Animal with jointed appendages, go to 6. Animal without jointed appendages, go to 5. Animal with long and cylindrical body, annelida. Animal with short, stout body, tremando. Animal with antenna, go to 7. Animal without antenna, go to 8. Animal with 1 pair of antennae, insecta. Animal with more than 1 pair of antennae, crustacea. Animal with pincer-like mouth parts, arachnida. Animal with sucking mouth parts, acarina. Animal with long ribbon-like body, cestonda. Animal with circular body, crinoidea. So, we come to the way you are supposed to mark. Question 1. Where you are given the materials provided, you identify the food substance, the liquid record, the food substances tested, procedure, observation, and conclusion. And so the food substance being tested was a starch, reducing sugar, protein, fats, or lipins, or oils. For starch, the procedure was you add a few drops of iodine solution to the liquid L. The observations got to a blue-black color. Starch was present. For reducing sugar, you add an equal amount of Benedict solution to liquid L in a test tube. Heat it to boil. Color changes from blue, green, yellow, orange. A tick. Reducing sugar present, a tick. Notice that for the procedure for reducing sugar, you are having two ticks. One of the tick is the procedure, and then the statement that you heat to boil. All right. Protein. To liquid in a test tube, and a few drops of 2% sodium hydroxide solution followed by 1% copper 2-sulfate solution. A tick. Purple color formed. Protein present. Fat or liquids or lipanes, you put a few drops of liquid L on the filter paper, warm to dry, a tick. Place against light, a tick. So in that procedure, you have two ticks. Permanent translucent spot formed. Fats, lipanes, oils, pleasant. Practical question 1B, marking. After stirring thoroughly, place 20 drops of liquid in the test tube and boil it. You find coagulation or formation of a solid or particle. Formation of a solid particle or card. What conclusion can be drawn from your observation in B1 above? Protein present. Marking question 2A. Name the parts labeled P, Q, R, S, and T. P is cell vacuum. Q is Golgi apparatus. R is plastid. S is mineral lamella. T is endoplasmic reticulum. Name the kingdom to which the organisms from which the tissue was obtained belongs. Plantae. Give 4 reasons for your answer in B1 above. Presence of cell vacuum, presence of cell wall, presence of mineral lamella, definite shape or regular shape. So, we have the specimen for question 2. And we have already naming the structures P, Q, R, S, and T. Cell vacuum, Golgi apparatus, plastid, mineral lamella, and the plasmic reticulum. The tissue is obtained from plantae. Reasons: presence of cell vacuum, presence of cell wall, presence of mineral lamella, and definite shape or regular shape. You measure X to Y and you get 16.2 centimeters. So let me go back. So this is X here. X is here. X. You draw a line up to Y here. You measure, and that is the range which we are getting as 16.2 centimeters. Centimeters will give you 162,000 micrometers. So magnification is equal to diameter of the image over action diameter, and therefore action diameter is equal to 62,000 over that 1,000, and it still gives you 5.4 micrometers. So, in each case, you are showing the sequence of the steps which were followed to arrive at the identity of the organisms. And for animal E, you had 1B, 2A, Arctic, you get Mollusca. F, 1B, 2B, 3A, 4A, 6A, 7B, Crustacea. You get 2 ticks there. 1B, 2B, 3A, 4A, 6B, 8A, Arachnida. 1B, 2B, 3A, 4B, 5A, Annelida. And J was 1A, 9A, tick, says Tonda. Thank you for watching. I hope you have found this practical session insightful. Be sure to try out these experiments in your own studies. If you have any questions, drop them in the comments. Don't forget to like, subscribe, and turn on notifications for more biology content. See you in the next video. And we acknowledge ChatGPT for coming up with the opening and closing remarks. We have the micrograph and photograph used here were sourced from NaOH, plus the questions. Finally, the slides were formed by me, Mr. Midamo TK. Goodbye and see you next time.